Oxytocin is a hormone synthesized in the hypothalamus and secreted by the posterior pituitary. It can contract cells around the breast and promote breast milk with lactation function, and has a strong promoting contraction effect on the uterus. The oxytocin signaling pathway initiates a response by the binding of oxytocin to the receptor. The oxytocin receptor belongs to the type G protein coupled receptor, or GPCR family, that can activate several downstream pathways upon ligand binding.
In the oxytocin signal transduction pathway, first we have the binding of oxytocin to the cell surface transmembrane oxytocin receptor that activates the G protein, G-alpha Q11. Then phospholipase C is activated, which hydrolyzes PIP2 to ionize P3 and DAG, ultimately resulting in the release of calcium ions from the sarcoplasmic reticulum and activation of protein kinase type C. G-alpha Q11 also causes activation of voltage-regulated ion channels and calcium ion entry into the cells. These ions then bind to calmodulin, and the calcium-calmodulin complex activates myosin light chain kinase, resulting in myometrial contraction. Both oxytocin receptor and protein kinase type C activate the mitogen-activated protein kinase cascade, or MAPK for short, resulting in increased systolic phospholipase A2 activity. Increased systolic phospholipase A2 activity and MAPK activation results in prostaglandin production, which also contributes to the contractile effect. The activation of the Rho-A to Rho-A associated protein kinase cascade by the oxytocin receptor is another pathway that results in increased myosin light chain phosphorylation and myometrial contraction.